Hello, everyone. My name is Mateo. I'm a fourth-year biochemistry major here, and my research is on investigating the effects of heat shock on the response of E. coli and Staphylococcus aureus to chloramphenicol and polymyxin B. So a little bit of background. Antibiotic resistance has become a major issue as bacteria are continually mutating to commonly used antimicrobial agents. And when researchers have been investigating what has been causing some of this resistance, heat shock proteins or HSPs have been implicated in some of this resistance. HSPs are proteins that assist in protein refolding after denaturation that's caused by stress. And when induced, these HSPs can have protective effects against this stress. And research has shown that high levels of HSPs, when produced in the presence of antibiotics and heat stress, can help bacteria survive. So for our research, our objective was to examine how heat shock affects the responses of E. coli and Staphylococcus aureus to antibiotics. And in this case, we chose polymyxin B and chloramphenicol. And our hypothesis was that bacteria grown at the heat shock temperature will produce HSPs and acquire some antibiotic resistance. And we will see that in a decrease in the efficacy of the antibiotics we used. So what we did was we took E. coli or Staphylococcus aureus from the streak plate, put it into a broth tube, and then incubated it overnight at 37 degrees. And then the next day, we took that broth tube, we took 150 microliters and put it into two new tubes. And one of those tubes was put into a 42-degree chamber where it was left there for 6 hours, and that was our heat shock treatment. And then or the other one was put into a 37-degree chamber where it continued growing. After 6 hours, the bacteria were plated onto an agar plate, and the antibiotics were introduced in the form of an antibiotic disc, and they were added immediately after plating. And then all the plates were put back into 37 degrees and left there for 4 days. And after 4 days, the zone of inhibition from the antibiotic disc was measured. So over here, these are the results from our plates. These are the plates from E. coli. These are the plates from Staphylococcus aureus. And CP stands for chloramphenicol and polymyxin B, BB. These were the plates from trial 1, trial 2, trial 3. And you can see there's 2 discs on each plate of the same antibiotic, and there's the zone of inhibition around it. So what we found was that there was no significant difference between the zone of inhibition for bacteria grown at 37 degrees or 42 degrees with either polymyxin B or chloramphenicol. And these graphs over here just show a comparison of the measured zone of inhibition for E. coli and Staphylococcus aureus for each antibiotic, chloramphenicol and polymyxin B at each temperature. The reason there's no blue bar or polymyxin B bar here is because Staphylococcus aureus is inherently resistant to polymyxin B. As you can see here, there is no zone of inhibition when polymyxin B is used. So it was 0, and for some reason the blue bar didn't show up because it's 0, but that's why. So overall, the temperature difference had no effect on the inhibition by the antibiotic. Fortunately, I didn't have enough time to do this. We weren't able to actually determine whether or not the heat shock proteins were actually present. So we came up with a few different conclusions as to why the results came out the way that they did. One is that we were operating under a simple model of more heat equaled more heat shock proteins, and it would just produce these heat shock proteins and they would just kind of linger around. That's not the case. It actually, as cells are, they're all just super complicated and everything has to be complicated. So they actually operate under a complex negative feedback system where it really, the number of heat shock proteins are in the cell depends on the number of unfolded proteins to folded proteins, and that's what determines the number of heat shock proteins that are around in the cell, and they don't just kind of like persist. And then also another potential issue was that 42 degrees might not be a large enough temperature difference. And maybe the amount of time to 6 hours that we used under heat shock treatment might have not been long enough to produce a strong enough heat shock response. And then finally, it's possible that the initial cells that produce the heat shock proteins might have been diluted out as the bacteria grew. So, when we treated it with the heat shock for 6 hours, then we plated them, and then they were left there for 4 days. The bacteria, as they grow, the ones that actually produce the heat shock proteins might have been diluted out, especially if we put it back at 37 degrees. But for future work, one is using larger temperature differences, probably more in the end of 45 degrees. Also, maybe monitoring the ratio of heat shock proteins to unfolded proteins during heat shock, just taking into account some to monitor how many heat shock proteins are being made throughout as heat shock treatment is being used. And then also maybe using a liquid antibiotic and measuring the survival in the broth tube. That's just part of the reason why it would be great to put it back into 42-degree chamber to sort of keep the heat shock treatment. But there was concerns over drying out the plates, especially after 4 days. So maybe using a liquid antibiotic and keeping the bacteria in the broth tube and measuring the amount of cells that survive in the broth tube might get rid of those concerns about drying out the plates, especially when using higher temperatures for long periods of time. And then finally, detecting the heat shock protein response presence. Again, I ran out of time doing this, but we were going to do an STS page experiment to demonstrate that they were there, or if they weren't there, or maybe if I had even more time, I would be able to do some sort of antibody experiment, but doing some sort of experiment to see if the heat shock proteins are actually present and are produced. And that's pretty much it. Thank you for listening, and I hope you enjoyed.